Welcome to Bio 205 Microbiology at Pima Community College. In this video, we're going to show you the serial dilution technique in order to quantify bacteria in a sample of carrot juice. I'm, for demonstration purposes, only going to show you a sample of our carrot juice here. In lab, we would certainly be also examining a certain amount of ground beef. So first, gather all of your supplies. You will need the following in order to successfully complete your experiment. You're going to need waste for your tips. We're going to have another waste bucket here. I'll show you as I walk through what these wastes are for. We're going to have what's called dilution blanks. This is essentially a salty, sterile water solution. And we're going to range our dilutions from 10 to the negative 1 to 10 to the negative 4. You guys are going to learn about the process of serial dilutions in the lab manual reading and as you work with your instructor. So for today's purpose, I'm going to focus on the technique and not the math. You're also going to need a sample of your 100% carrot juice or your ground beef, whichever one you're working with at the moment. You're going to work with what's called a micropipette. We have this set here at 500 microliters or half a mil. You're going to need tips for your micropipette. It's really important that you have a vortexer on hand. You're going to be vortexing a lot as you work through this experiment. Here at Northwest Campus at Pima, we have pre-sterile plastic spreaders that we're going to use to rub the bacteria all over the Petri plate at the end. Some campuses use glass and would use the alcohol and flame it. You're going to have several Petri plates here, so for the interest of time, I've already labeled these ones for us. We have two types of media that we're working with. One is called trypticase soy agar. It's the light yellow one. We'll learn about this in the lab manual. It's general media, non-selective. And because we're examining carrot and beef from our background reading, we know that we're going to be looking for what's called coliform bacteria, bacteria that come from the gut. And so this could be present in our carrot or in our carrot juice, could be present in ground beef. We'll learn about that later. But DLA is a type of media we learned about before, and this is going to select for the gram-negative rod-shaped bacteria that we find in the gut of mammals. So we have several different dilutions, and again, we're going to read about the process of going through this, but we have essentially a trypticase soy agar for 10 to the negative 1, 2, 3, and 4. And for the DLA, since it is selective and it's going to kill off a certain population from our sample, we're only going to explore the first two dilutions, 10 to the negative 1 and 10 to the negative 2. So we first, depending on the lab manual, there's different ways that we can do this, but we're going to first start with our serial dilution. So to perform your serial dilution, you first want to place a tip on the end of your micropipette, like so. Gently push down to make sure that the tip is in place. It's generally decent practice to shut the lid. We're going to take our sample of carrot juice, just invert it to gently mix it really quickly. And we're going to take out 500 microliters or half a mil from our sample. I'm going to push down to the first stop on my micropipette using my thumb. Place the tip into the liquid and gently release the plunger of the micropipette. I'm going to go into my first dilution, which is 10 to the negative 1. I'm going to enter the tube and push down to the second position or the second stop on my micropipette. Place the cap, dispense the tip. Now we're going to take our test tube, securing the lid in place, the cap in place. We're going to now vortex our sample to mix the sample. About 5 seconds should be sufficient. Now we're finished with our carrot sample, so I'm trying to push this out of our field of view. It's really critical that you replace the tip with a fresh tip. Now using our 10 to the negative 1 dilution, we're going to remove a tenth of our population of cells, and we're going to place it into our second dilution tube, labeled 10 to the negative 1. So I have my pipette and my pipette tip ready. I'm going to remove the cap with my little finger of my dominant hand, pass through the flame, go down to the first stop on my micropipette with my thumb, enter into the tube, and gently release the plunger. Release to the second position on the pipette to release all of the liquid. Place the cap. Dispose your tip. And again, repeat your vortex. I'm going to move my test tubes out of the way as I work to know that they are complete. And now we're going to repeat our serial dilution by taking 1/10 of our population from 10 to the -2 and moving them over to the 10 to the -3. Ideally, you should not see any air bubbles in your sample when you pipette it. Vortex. And repeat this dilution one more time. Now we're going to go from 10 to the -3 to our final dilution, 10 to the -4. So we're removing about 1/10 of the population of our cells and moving them next door to this dilution blank. Vortex. Notice when we're finished with our serial dilution, I'm only showing you 3 out of the 4 tubes here, but notice that just by looking at the carrot juice alone, I can tell that I have successfully diluted my sample because the color becomes lighter and lighter with each dilution tube. That is also representative of the bacterial cells in your sample, even though at this stage you can't visibly see them with your naked eye. Once your serial dilution is complete, we're ready to inoculate our bacteria onto our Petri plates. So in order to add our samples to our Petri plates, you want to first make sure your Petri plates are labeled. We have our 4-trypticase soy agar labeled 10 to the -1, 2, 3, and over here the 4th one, 10 to the -4. We have our 2 DLA plates lined up, so this is 10 to the -1 and 10 to the -2. Notice that I have my lids on top because I'm going to be prepared to add my sample quickly and to spread the bacteria out before it dries. So if your plates are labeled and you have them lined up, you're going to now be prepared to add your sample doing the following. Add a fresh tip to your micropipette. Pick up your first dilution tube, which is 10 to the -1. Make sure to vortex this before adding your sample. We're going to remove the lid. And this time, instead of 500 microliters, we're going to only take out 100 microliters. This is essentially another 1/10 dilution. We'll see how this math works out later. Go to your first Petri plate, remove the lid. Use a finger to stabilize the pipette, float above the surface of the agar, dispense down to the second notch on your plunger. Place the lid down. You're now going to dispose of your tip. And before moving on, you really want to make sure that you spread the bacteria out evenly across the plate because you guys are going to be counting colonies, especially from images, so we want to make sure we do a good job spreading it out. The longer you wait, the faster or the more likely your bacterial sample is going to dry and you're not going to be able to get a decent count. So we're going to remove a sterile inoculating hockey stick. At least that's what it looks like. You don't have to sterilize these, just make sure you don't touch it onto the table, don't grab it with your hand, try not to breathe on it. You can remove the lid, place the spreader down into your sample. With one hand, my left hand here, I'm going to rotate the Petri plate in a circular motion. And with my inoculating hand, I'm going to use the spreader to literally spread the bacterial sample out over the entire surface of the agar. We only use 100 microliters here because if we go up to even half a mil, it's going to take a really long time for your sample to dry. 100 microliters is the sweet spot. There's no limitation on how long you really have to do this, but generally speaking, more the better. The more dry your sample becomes, the better. Take note though, the longer you have your lid off of your Petri plate, the more likely you are to contaminate it from the air or from your breathing. Place the lid back on. I'm going to flip it over and put it out of my field of view here, just so I know that it's been inoculated and it's ready for incubation. It's really important I dispose of my spreader. And now we're going to repeat this process for the next Petri plate for 10 to the negative 1 on our DLA. Don't forget to vortex your sample. Take a new spreader and repeat the same process. For demonstration purposes, I'm going to speed this up just a bit, but again, we would do this until the Petri plate is ideally dry. Repeat for the next dilution. Now we're working with 10 to the negative 2. Making sure to vortex my sample between each plate and always take a new pipette tip. Really doesn't matter which order you do this in, as long as you're being very certain of which tube you're taking your sample from. If they get stuck, you can simply remove it. If your glove becomes soiled, though, you might have to change it out. We're still working with 10 to the negative 2. We're now done with our DLA, so we're just going to finish up here with our TSA samples. Again, the longer the better. You want your plate to be dry. We have 2 more samples to go. Now we're at 10 to the negative 3. You want to try not to gouge the agar, though sometimes it's really difficult not to scratch the surface. It's okay. Try and cover the entire surface area of your plate as you inoculate it. And finally, our last sample, 10 to the -4, which we have in order. We need one final pipette tip. It doesn't look like much, we can't see orange coloration, but don't assume you don't have any bacteria in your sample. Your plates are now ready for incubation. When you come back to the second class period, for you guys virtually, if you are virtual or if you are online, you're going to be viewing images of our final results there. You will be counting the number of colony-forming units in order to calculate the number of bacterial cells that we had in our original sample of carrot juice. So that's the basics of the serial dilution technique. One variation you might see between campuses is some of us use micropipettes, others use the serological plastic pipettes, but it's going to achieve the same outcome. Have fun!